I have to admit that my presentation will be mainly on hepatitis C, because the work we are conducting is under a grant from a donor, specifically on hepatitis C, but the technology we are developing have been designed with a polyvalency in mind. So all these technologies, the majority of them, can be adapted for other viral hepatitis assays or even other assays. I will give you some examples during the presentation. So first of all, I will give you a brief introduction about FIND, and then I will guide you through our work in product development, and then there will be a few other slides on other groups that are developing a point-of-care additive for liver function test. So FIND is a non-for-profit organization with the main goal of catalyzing the development of diagnostic tools for poverty-related disease. And diagnostic tools are not just machines, equipment, are also complementary devices that can improve the quality of the testing. Very recently, we launched a micro device for transferring the same amount of blood on a rapid test in order to improve the pre-analytical phase of the rapid test. We cover several areas, and very recently we added antimicrobial resistance and outbreaks, two areas where the diagnostic component is highly neglected. Our work is built around 4 pillars. In addition to catalyze development by supporting companies to develop fit-for-purpose products, we also work with WHO and other international organizations to design and implement clinical trials in order to generate evidence necessary for a policy guidelines improvement. We work with governments in order to accelerate the uptake of the new products. And finally, we lead several discussions globally around the agenda on diagnostics. So we are very active in all the 4 pillars across the several diseases. We are based in Switzerland. However, we also have 3 offices in South Africa, India, and Vietnam. Very recently, we closed the office in Uganda, and we are planning to open an office here in Kenya, hopefully soon. As you can see, so FIND is a product development partnership, and in its nature, the PDP is capable to collect different resources, not just financial, but also knowledge and expertise from different players, and put all these resources on one single project in order to increase the likelihood of being successful. So these are the type of players and partners we work with across all the diseases. So that's all about FIND. So what is a public health approach? We probably are all familiar with the elimination targets established by the WHO, approved by the World Health Assembly, of eliminating hepatitis B and C by 2030. So elimination means a reduction in incidence and a decrease in mortality. So we really want to be sure that if we embark in a public health approach, we are doing it in the right framework, with the right guidelines and policies, using the right tools. So in terms of hepatitis C, the testing algorithm has been released by WHO in 2017. As you can see, is a simplified algorithm where there is one screening test, one confirmatory test, then there is liver stage, and a test of cure at the end of 12 weeks after the end of the therapy. However, even if we have a simplified testing algorithm, the tools we are supposed to use to scale up a public health response to hepatitis C are not yet there. And this is mainly related to the quality of the products that we can access to the market, and the capacity of being used in a decentralized setting. But also the lack of understanding from companies and also donors on the magnitude of an hepatitis C market is a huge barrier. So the first activity FIND conducted last year was to assess a potential need and demand for hepatitis C diagnostic tools in 29 countries. This report is available on our website, has been shared with companies as a way to stimulate investments from their end. there are several needs that are not met across the entire health system in regard to viral hepatitis testing. So these needs are, some of them are in need, of course, of new technologies. There are not the right technologies to be used at community level or health one level facility. But there are some needs that can be addressed by improving the efficiencies across the health system and optimizing the allocation of the resources available. And this is very important because viral hepatitis, as we heard over the last couple of days, very likely cannot rely on funding coming from external donors. So it should be really part of a national budget. So it is in this context of lacking of quality assured diagnostics that back in 2016, FIND, with the financial support from Unitaid, established the hepatitis C elimination through access to diagnostic program with the objectives of accelerating development of diagnostics, improving access to these diagnostics, and establish evidence by piloting projects in 6 different countries for informing policymakers on how to advance access to hepatitis C diagnosis. So, for the purpose of this presentation, I will only focus on our R&D work, but we have a huge program also in implementing these pilot projects in 6 countries to prove the efficacy, to prove the feasibility, the effectiveness of these tools in a real setting. So we are focusing on 3 areas. One is looking at improving the current infrastructure to decentralize HCP diagnostics. The second one is looking at developing game-changing point-of-care solutions. And we're also developing a serology program aiming at increase access to hepatitis C screening. I want to point out from this slide, if you look at the column FIND work, so our work is really across the continuum of product development. So FIND is involved in feasibility studies, test development and optimization, up to the implementation of clinical trials that are designed with in mind the requirements of several different regulatory agencies. And this is important. So our work is not to do clinical studies for the purpose of doing clinical studies. We want to help companies, put them in the conditions of minimize the burden of doing clinical trials in high burden countries where otherwise companies cannot be successful. So let's start with the first area. We had a first project that finished early this year. Started in 2017, was a multi-center clinical evaluation of a HCV RNA cartridge developed by CFAID, using as a sample whole blood coming from a finger stick. As you can see from the table, the performance is very good, both against the Abbott machine and against the Expert plasma cartridge, which is already available on the GeneXpert. This study was conducted in Georgia, Cameroon, Greece, US, Malaysia, to have a diversity of genotypes, but also Greece to allow the company to have a claim for a CE IVD mark. And that, as you can see from the procedure, is extremely simple. There is no venous blood collection, which means is very well indicated for those patients where a venous blood collection might be extremely difficult. And then if you think about hepatitis C, high-risk groups, people who inject drugs have the highest burden of hepatitis C. So this is likely to be very well adopted and successful in increasing the acceptance of this high-risk group for NHCV RNA. The second project we embarked on is a collaboration with GeneDrive, also near POC for HCV RNA. We are doing a validation of this platform in real settings. It's an independent evaluation, and we really want to bring the device where it is supposed to be used. So we don't want to go in a central lab in a tertiary facility where everything looks nice. We really want to go where patients have access to the health system and prove it that is user-friendly, can have an impact, can reduce the loss to follow-up if also screening is available on site, and other operational research questions. The main difference is that there is a need for plasma. And there are some manual steps that are not in the fingerstick cartridge. The last activity in trying to improve the utilization of the current resources is a validation study looking at assessing the clinical performance of dry blood spot for HCV RNA. And this study is done in close collaboration with 3 commercial companies who validated internally the DBS protocol, gave us the protocol, and we designed the study in a way that the data generated by this study are accepted by the WHO pre-qualification team for a dossier submission from the company. So as you can see, all these elements are crucial to accelerate not only the development, but also the access to. There are many studies that are designed without having the clear WHO-PQ as an endpoint. There are studies that are not properly powered. So what we are doing here is to really streamline the process from internal validation to regulatory submission. So this study will take place in different countries, in 3 countries. However, we are working in the in-country pilot projects. We are already working with the National HIV Program and the National Hepatitis Control Program to make sure that we start the discussion around integration by piloting the use of plasma samples on the existing high-throughput platforms available in tertiary health facilities. By doing this, once the DBS protocol has been validated, it will be much easier to introduce in the workflow because we already did integration through the plasma samples. So the second area, the orange one, is the development of game-changing point-of-care solutions. And to minimize the risk of this very ambitious project, we decided to target 3 different types of products. The first one is a molecular test. We really want to make sure that we have a low risk by embarking in a molecular test development with the probability of having high sensitivity, high specificity. However, since we are talking about, again, public health approach, we want to keep the costs low, final product costs very low. And the disadvantage of this approach is that very likely in comparison to the other 2 approaches, the final cost of the goods will not be as low as the other 2. The other approach is to develop an immunoassay platform, in this case specifically for a core antigen test, that will likely be lower in terms of cost of goods. However, I can tell you that there are more technical challenges for this kind of assay in comparison to the molecular platform. But if you look at the advantages, so we are putting resources to develop a sensitive immunoassay platform that can be used in lower level health facilities later on also for other assays. So we can target the different markers for hepatitis B, a P24 antigen test, for early infant diagnosis. So this is a very important project. Even if will not be successful for HCV core antigen, having the platform available is a big step forward for leveraging to get additional funds for other diseases. And then there is a dual test for antibody antigen, HCV antibody antigen, in a lateral flow assay format that is the lowest in terms of costs. However, also in this case, the risk of having a suboptimal sensitivity are very high. But if you think at the potential, at the 4 results you can get from this test, you have antibody negative, antigen negative, in this case is negative, antibody positive, antigen positive, in that case is positive, so you don't need a confirmatory test. If antibody positive, antigen negative, you still need a confirmatory test, but in a much smaller proportion of the population. And in case antibody negative, antigen positive, probably is a wrong result. So these are the 3 projects we are embarking in developing disruptive point-of-care technologies. The process was pretty long, so we called for an expert meeting back in 2015 to develop the requirements, part of a TPP, target product profile, that are listed here in this table, and the TPP was published last year. Following the publication of the TPP, we put out a request for proposal to invite companies to apply for this project. And we received more than 30 applications, so that's a very encouraging, meaning that there are at least 30 plus companies worldwide interested in a POC approach for infectious diseases. I have to say that TPP, these are not strict requirements, it's more a guidance for companies to make sure they target these requirements in developing their proposal. So the evaluation of the applications was a very extensive work because of the good quality of the applicants. And in quarter two, in July this year, we announced the award of the grants, and we embarked in 8 feasibility studies. We are working with Abbott, Blink Diagnostics, and Diagnostics for the real world for a molecular point of care. We are working with ChemBio Diagnostics, Mologic, and DCN Diagnostics for a core antigen rapid diagnostic test. And we will work with 2 additional companies to be known by the end of September to develop a high-sensitive immunoassay for HCV core antigen. So the last area where we work is serology. We are embarking in 2 projects here. The first one is looking at improving the number of WHO pre-qualified RDTs available for procurement by international agencies or national governments. We are doing this in a stepwise approach. The first study we are conducting is a performance evaluation of 10 RDTs that are commercially available but don't have any stringent regulatory approval, meaning we don't know the performance of these tests. We don't know how good they are. So we are doing this study in Nigeria and in Europe. It's a retrospective study looking both at mono-infected and HIV, HCV infected patients. This is a research question that WHO asked us to answer. And then the next step will be to work with the companies that have the best performers, RDTs, to improve their performance, so to reach the point where they can submit the dossier to WHO pre-qualification. At the same time, we work with the companies that are interested in developing a combo test, a combination of HIV, HCV, HCV, HBV, or HIV, HCV, HBV, has to be decided, as a way to, again, improve the availability of RDTs that have meet quality assurance requirements, but can be used in different settings. And then we will work also with those companies that are interested in developing a HCV RDT self-test. As you probably know, HIV self-test has been rolled out in several countries, and we think, together with WHO and other international stakeholders, that an HCV self-test can find its space in the response to eliminate hepatitis C. Last but not least, we are also working on a DBS sampling for enzyme immunoassay. This is a company who already has a WHO pre-qualified ELISA test. We are clinically validating the performance of the assay on a DBS because, from a programmatic point of view, if you have a DBS card where you can do HIV viral load, HCV RNA, and HCV antibody, when you have patients coming for monitoring for HIV viral load, with the same card, they can get the HIV viral load, screen for HCV, and if positive, you can use the 3rd blood spot for a confirmatory test for hepatitis C. So in terms of optimization of resources, is a very good project. The timelines, I have to admit, are ambitious. but we are pretty sure that, at least in each of, at least one good product out of the yellow projects will come out in the next 2-3 years. So this is all about find work. So as you can see, we have approximately 15-16 projects going on, with the goal, again, of establishing, piloting the acceleration of viral hepatitis products as a way to bring in other viral hepatitis soon. So we already started exploring the possibility of including HBV DNA in one of the point-of-care solutions. So, as we heard from Mark Turst the other day, he mentioned this TreatB score. I didn't know about it, honestly. So I did some research, and it's actually, very interesting, because when we talk about innovation, we always think about, fancy technological, devices, or using connectivity to transfer data from a remote place where there are RDT. These are all important tools we need to put in the basket. However, innovation is also in the way we use the current tools. And this TreatB score is fascinating because it uses HBe antigen and ALT as a way to score the eligibility of patients for HBV treatment. This was, I think, a preliminary study. There are other groups doing piloting the TreatB score in other contexts, so we need to gather stronger evidence. But again, if you think from the public health lens, is a very cost-effective approach. So I did some research in this regard, and I found out that there are 3 potential RDTs for ALT function. So one is developed by Burnett Institute in Australia. They have a spin-off company in China. And I saw the device. I mean, it's a rapid test like malaria or HIV. And I spoke with them the other week. They told me that they are planning to do a clinical validation of this device in Q4 this year. So hopefully by the end of next year, we'll have robust data for regulatory approval. The other company is not really a company; it's a non-for-profit organization called Diagnostics for All, based in Boston, also a spin-off of Harvard University. And this is a low-cost. This is very intriguing because it's a paper-based, so it's big like a stamp. That can be used for assessing the level of ALT in whole blood. So they are moving forward with the FDA registration, and hopefully, FDA can take quite a long, but hopefully also this device will be available for the market by the end of next year. And then I found a paper developed by a group always from Harvard, where they try to put together to combine ALT and AST. But I mean, this is only still at the research level only. It's important to note that an ALT test is not just for assessing the liver staging in a public health approach for hepatitis C, but can also be used for monitoring the toxicity of some drugs, especially in HIV and TB programs. So there are several applications of a rapid test for the liver function, for ALT, sorry. So we are at the stage of, at the stage of embarking in this massive product development, but we are fully aware that new products will not have an impact if they are not integrated into a comprehensive package for scaling up a public health approach for hepatitis. And this package includes at least 4 elements. So one is the diagnostic, of course. The other one is the treatment. The most important one probably is the advocacy, so raising the awareness. As we heard yesterday multiple times, there is no awareness among patients on the risk of hepatitis C, or the importance of getting tested, of the importance of getting cured. So awareness, diagnostic, treatment, and programs. So we need to have national programs with the right budget, with the right champions in it, as we heard from Professor Manal the other day, that can lead the way to viral hepatitis elimination in each country. I want to make a comment about the pilot projects. So from my experience in HIV, there are hundreds of nice pilot projects looking at different service delivery models, targeting different populations, innovative ways of providing, different services. But how many of them have reached a scale-up level? Probably very few. So it's very important that when we design a pilot project, we have the end in mind, which is the scalability of the model we want to implement. So we're using this approach to design the pilot projects in Cameroon, Georgia, India, Vietnam, Myanmar, and Malaysia. And even if there is only one African country, I can tell you that the models we are piloting in these countries, if proven to be successful, can be replicated in other countries and for other diseases, specifically hepatitis B, also here in the African continent. What is very important when we look at the utilization of these tools is there are 4 pillars we need to take into account. So the one is the simplification of the testing algorithm. We have that. WHO is giving us some guidelines on how to simplify the testing algorithm. The second one is to decentralize the services. As a public health approach, the focus is on population. So we need to answer the needs of the communities. We need to have target interventions to answer these needs. The third one is integration. So without leveraging the huge success achieved by HIV and TB national programs, I have the impression that we are not going too far by eliminating viral hepatitis. So we need to take into account that integrating a response to viral hepatitis into the existing HIV and or TB infrastructure is key. The fourth one probably does not apply that much to hepatitis B, but for C is key, is differentiation of care. We really need to take into account that high-risk groups for hepatitis C behave completely different. In one of the countries in Georgia, we are piloting the screening confirmatory tests in 3 armed action sites. Before our intervention, there was a drop-off between screening and confirmatory of 50%. Patients didn't want to go to the next level for HCV RNA. Now we are down to 2%. So even if just preliminary data we started in May, we only have 300 patients so far. So I think it's encouraging to see how the deployment of the right technology in the right setting, targeting a specific need of a specific group, can be an effect also in the short run. So I want to thank you again, the organizers, and I'm open for any questions. Thank you.